Welcome to this explainer. Today, we're going to build a massive mental map of the brain's associative learning mechanics. We are zooming all the way from macroscopic, everyday behaviors, down to the absolute deepest, most fascinating molecular secrets of the brain. So, if you've ever wondered exactly how your brain physically learns to link two completely unrelated events together, you're in the exact right place. Let's get into it. Here is our roadmap for today. We'll quickly cover the basics of associative learning, mapping the brain, cellular secrets, memory reconsolidation, and finally, integrated systems in action. Starting with Section 1, the basics of associative learning, setting the foundation. Okay, so we've got to set a quick baseline for how our brains link events together without any conscious effort on our part. To really understand this, we need to look at classical conditioning. This is a crucial component of the non-declarative or implicit memory system. What does that actually mean? Well, it means your brain is establishing a predictive relationship between two events completely behind the scenes. The key takeaway here is that this type of learning happens totally under the radar. It forms associations and operates without us even trying to actively remember them. It is quite literally a fundamental biological survival mechanism. Think about exactly what is being linked together here. We start with a conditioned stimulus, or CS. This is an initially neutral event, like, say, hearing a simple tone. On its own, it means absolutely nothing to you. But then we introduce the unconditioned stimulus, the US. This is an event that naturally evokes a measurable, often pretty intense response. Think of a mild electrical shock. By pairing a totally meaningless signal with a highly significant biological event, the brain learns to predict what's coming next. Suddenly, that neutral tone becomes a blaring warning beacon. Moving on to Section 2, mapping the brain, amygdala, and cerebellum. The brain brilliantly divides the labor of learning. On one side of this equation, we have the amygdala, which acts as the ultimate hub for learned fear and emotional associations. On the flip side, we have the cerebellum. This is the critical structure for conditioning your skeletal musculature and refined reflexes. This hard distinction is a really vital pedagogical anchor for us as we dive deeper today. Your amygdala handles your emotional conditioning, while your cerebellum handles your motor conditioning, like learning to reflexively blink your eye at a tone that predicts a puff of air. To really teach this in depth, let's trace the actual anatomical pathway of fear right through the amygdala. Step 1 is sensory integration. Sensory data, whether that's a sound, a sight, or a touch, enters the basolateral nuclei of the amygdala. This is where the initial pairing of, say, the tone and the shock creates physical synaptic changes. Then, in Step 2, those signals travel to the relay point, which is known as the central nucleus. Finally, we hit Step 3, coordinated outputs. The central nucleus sends commands directly to the hypothalamus to trigger autonomic responses, like spiking your heart rate via the HPA axis, and to the periaqueductal gray matter, or PAG, to trigger behavioral reactions, like freezing perfectly in place. It's literally an incredibly efficient, hardwired circuit. All right, Section 3, cellular secrets of synaptic plasticity. This brings us to a foundational concept in neuroscience, famously summarized by Donald Hebb. Neurons that fire together, wire together. This famous Hebbian modification rule explains that for conditioning to actually physically occur in the brain, the synaptic activity has to correlate strongly with a postsynaptic response. If the tone neuron and the shock neuron are firing at the exact same time, the physical connection between them gets a massive upgrade. But here is the multi-million-dollar question. How does the neuron actually know they are firing at the same time? The answer lies in molecular coincidence detectors, specifically NMDA receptors. For the neural connection to strengthen, you need, Step 1, the presence of glutamate from the presynaptic neuron. But that's not enough on its own. You simultaneously need, Step 2, the postsynaptic membrane must be sufficiently depolarized. Only when both of these specific conditions are met do we get Step 3, where calcium is finally allowed to enter the cell. I want you to think of NMDA receptors as a secure bank vault that requires two distinct keys to be turned simultaneously just to let calcium rush into the cell and rewrite the synapse's strength. Now, this rewriting process can actually go in two directions. Long-term potentiation, or LTP, is induced by high-frequency activity, which strengthens the synapse. Conversely, long-term depression, or LTD, is induced by low-frequency activity, which actively weakens the synapse. And this brilliantly illustrates the difference between our two brain regions. While the amygdala primarily relies on LTP to strengthen those fear pathways, the cerebellum actually uses LTD. In the cerebellum, you've got simultaneous inputs from climbing fibers, which carry error signals, and parallel fibers carrying sensory data, converging right onto Purkinje cells. This precise depression of synapses is exactly what creates flawless, refined motor learning. To prove just how fundamental this biology is, we can look at the sea snail, Aplysia. It uses serotonergic modulatory neurons to facilitate reflex conditioning. The cellular mechanism here involves increased cyclic AMP and protein kinase A, which ultimately results in increased neurotransmitter release. I really want you to take this in for a second. Even simple invertebrates use this incredibly complex chemistry to modify the strength of their neural connections. Learning is an ancient, molecularly conserved mechanism. It's just incredible. Section 4. Memory reconsolidation and malleability. For a really long time in neuroscience, we assumed that once a memory was consolidated into long-term storage, it was static. Like a book placed on a shelf, we thought it was just locked in forever. But what really happens if we recall that fear later? The reality, as modern research shows us, is far more dynamic and, frankly, way more fascinating. The process begins with step 1, memory reactivation. When the conditioned stimulus is presented again, the memory is recalled. But this recall triggers step 2, a temporary sensitive state. The memory actually becomes unstabilized. It has to go through step 3, reconsolidation, where the memory is fixed or saved again into the brain. Merely remembering something reactivates the memory, making it super vulnerable to alteration right before it saves again. Basically, every time you remember something, you are actually rewriting it. And this biological window of vulnerability has massive implications. Clinically, scientists are using this exact reconsolidation window to intervene. By administering drugs like propranolol or utilizing genetic switches like optogenetics in experimental animals, researchers can target a memory while it is in that temporary sensitive state. They can actually weaken the emotional impact of a traumatic experience or even introduce entirely false memories before the brain finishes saving the file. They are quite literally exploiting the mechanics of memory itself. Let's bring it all home with Section 5, Integrated Systems in Action. Let's look at the absolute most famous example of classical conditioning, Pavlov's dog. First, the sensory systems detect the auditory bell and the visual or olfactory cue of the meat. Next, the limbic system and hypothalamus step in. The amygdala processes the emotional significance of the bell, while the hypothalamus triggers the autonomic response, the physical salivation. Finally, the cerebral cortex provides higher-level analysis and the emotional coloring of the whole experience. When a dog drools at a bell, all these microscopic coincidences, those NMDI receptors and macroscopic pathways are firing in parallel across multiple integrated systems to produce one single learned behavior. As you consider the mechanics we explored in this explainer, all the way from the cellular coincidence detectors to the distinct roles of the amygdala and cerebellum, I'll leave you with this final thought. If reconsolidation literally allows us to rewrite associative pathways using drugs or genetic switches, what are the ethical implications for modifying human traumatic memories? Mastering the biology of memory gives us profound, perhaps even terrifying clinical power. Just how much of our past should we be allowed to rewrite? Thanks so much for exploring this with me today.